Howdy, and let's pick up on the first protein purification step, salting out. Now, in salting out, we're actually going to separate our protein, which is this green target protein, from contaminating molecules, the red and the orange. If you're colorblind, I apologize. Within this soluble fraction separated from our bacteria culture. So if we grew bacteria cultures in a 6-liter flask, we use centrifugation to separate the bacteria, which will pellet at high centrifugal force from the media, which is liquid, and you can dump that out, resuspend in an appropriate buffer, and lyse or break open our cells, spin again to separate the husks, the shell of the bacteria, from the cytoplasmic contents. And that's what's in our first test tube. To this test tube, we're also going to be adding a salt, typically ammonium sulfate or ammonium acetate. These are large ions which don't perturb the structure of the protein, but affect its solubility by competing for the water. Water will want a hydrogen bond to the ammonium ion and the sulfate ion when these salts dissolve in water. And that changes the solubility of our protein. And so we start typically with about half molar, 0.5 molar salt, ammonium sulfate, and we will, the first time we've tried purifying this protein, let's say it's insulin, we don't know what concentration of salt is required to change our protein from the liquid phase to the solid precipitate at the bottom of the tube after centrifugation. So again, these tubes are put into a rotor, into a centrifuge, centrifugal forces applied that will pellet large things and leave soluble things in the supernatant fraction. The first time you do this, you don't know whether your sample, your favorite protein, the green protein, will be in the supernatant as shown here or in the precipitate, the pellet. And so you save both the supernatant fraction and the pellet fraction in each step, and you move it to a clean tube, and you would resuspend this pellet to the same volume, and you would assay whether your sample was in that pellet or whether it was in the supernatant fraction. Okay, in this case, it was in the supernatant fraction, so a second addition of salt, and sometimes you have to go up to 10 samples before you figure out what concentration of your ammonium sulfate you need in order to precipitate your protein. After you've done it once, now you know that will be reproducible, and from then on you can use what you determined through your assay to be the appropriate salt concentration for salting out your protein. So once you've recovered the precipitate pellet and discarded the supernatant using an assay to make sure that you are not throwing out the baby with the bathwater, you can then resuspend your precipitate into an aqueous buffer that is at a pH that keeps your protein folded. So that's what we're going to be doing in the salting out step of separating our proteins during purification. Now, it turns out that proteins are least soluble at their isoelectric point. The isoelectric point is abbreviated lowercase p, capital I, and it is that pH at which the net charge of your polypeptide is 0. I'll say that again. The pH at which the net charge, all the pluses and the minuses cancel out, and you get a net charge of 0, that pH is the PI, or isoelectric point. And you saw this on an earlier slide in an earlier video, that there are PIs known for proteins. This is something that is automated now, and you can use an algorithm to calculate the PI of your protein, or you look up your protein and that will just be a part of the stats given for you. So from what we know about the pKa's of the groups that are ionizable on our amino acids, and now the fact that the polypeptide is formed as a linear polymer of amino acids, we know that we need to take into account the side chain pKa's for those amino acids with ionizable side chains, but also the alpha amino group's pKa, which is what? 9, that's right. And the pKa of the alpha carboxyl group at the C terminus, whose pKa is what? That's right, 2. So with those pKa's in mind and the pKa's for the ionizable groups on the side chains of amino acids that have ionizable side chains, you can figure out what the net charge is for your polypeptide at a given pH. Remember the mantra, if your given pH is below the pKa, protonate. If your pH is above the pKa, deprotonate. And if you have that mantra memorized, you remember your groups, for example, nitrogen-containing groups will be positively charged, a +1 when protonated, and oxygen and sulfur-containing groups will be a -1 when deprotonated, then you'll be able to add up the charges. And I have some practice problems for you to do that and check yourself on eCampus. The effect of this is that once you've figured out the isoelectric point, and we'll do a calculation to show you how you can actually calculate an isoelectric point, any pH that's below the isoelectric point will result in your protein carrying a net positive charge, but any pH above your isoelectric point will give your protein a net negative charge. Let me say that again. Below the pi, your protein carries a positive charge, and above the pi, your protein carries a negative charge. And that's because groups that are deprotonated above the pi that are going to be charged are the acidic groups. Those oxygens and sulfate groups are going to be, sulfur groups are going to be carrying a -1 charge. Whereas when you deprotonate an amino group, it's neutral charge. Using that logic, you can also convince yourself that below the pi, when you protonate groups, the only charge that's going to remain is a positive charge. So look for examples on eCampus for how to calculate the net charge and how to, given your protein sequence, how to calculate the isoelectric point. It's also outlined in the textbook. We'll begin with chromatography in the next video.